So it is my very, very great pleasure to introduce one of our own, Andy Stewart, who is the director of the Diabetes, Obesity, and Metabolism Institute here at Sinai. In fact, the founding director, and he has built this institute from nothing into really a major force within Sinai and now within the country in diabetes research, with a very translational focus on bringing novel therapies to humans in the area of beta cell regeneration. So Andy got his MD at what used to be called just Columbia University College of Physicians and Surgeons, and now, like every place else, has somebody's name attached to it. And I was a few years behind him, 4 or 5. But I remember Andy because he also did his residency in internal medicine at what used to be the Roosevelt Hospital. And I remember him as a resident. I think I was a medical student, and he was very smart then, and he has continued to be so. He then went to Yale for his endocrine fellowship, where he stayed for many years, rising to the rank of full professor with tenure. And he used to be what's known as bonehead, and was a leader really in bone metabolism. And one of his areas of interest was tumor-mediated hypercalcemia. And his group was isolated the responsible factor, PTHRP. And it was through working in this field that he discovered that it had a role in beta cell biology, which led to sort of a mid-career change and path to become a beta cell scientist from bone. And he went from Yale to be chief of the Division of Endocrinology at Pittsburgh, again, where he built this division into a major force, and had a sideline, which is very, very interesting, in workforce issues related to endocrinology. And I think was largely responsible for the fact that, in particular, diabetologists now are much more valued for their role in bringing important patients in terms of generating income, unfortunately, into the healthcare systems, and really increase the salaries for those physicians, and it's had a halo effect. Many of the others of us who were division chiefs at the time were always upset with Andy because he would just pay so much more than everybody else. And we sort of burned his letters when he was recruiting people. But I think that is an accomplishment that he deserves to be recognized for on the positive side of valuing endocrinologists. Then he came to Mount Sinai from Pittsburgh, where he has created the Diabetes, Obesity, and Metabolism Institute. And he's going to talk to us today about really exciting research on human beta cell regeneration and drug therapy that is work he's done here. So, Andy?
So thanks, Andrea, for actually a really personal introduction, more than I was expecting. So you can tell Andrea and I go way back. She was my medical student when I was a resident, if you can believe that. And actually, for those of you who are younger and don't know me as well, it probably comes as a shock that I was ever a real doctor. But we set the financial model in Pittsburgh for physician compensation in academic medical centers. And that got imported to New York and a bunch of other places. So I'm actually proud of that. To be honest, Andrea, I totally forgotten about that. I was like, well, where's she going? But so yeah, so thanks for that. So look, I'm going to tell you a story that, a series of stories that you've heard before in part, but some of it at the end, starting in the middle, is actually very new. And basically the take home message is, at Mount Sinai in your midst, we've discovered and are translating clinically the first classes of drugs that are able to make human beta cells regenerate. So I'm going to do this in a series of stories. Emily, I think if somebody has questions and they're quick, please interrupt me. If it looks like it's a longer one, let's save them for the end so we can finish.
Sure.
Okay, and there you have it. And also, you know, I don't know if you've been teaching medical students, but they actually request that the students leave their cameras on so the person who's talking can actually realize they're talking to some human beings instead of a black screen. So if you're comfortable leaving your screen on, it's more fun for me if I can actually see you. There you have it. So here we go. So the first story I want to tell you is a review. Many of you have heard this before, but I want to talk about beta cell mass and type 1 diabetes and type 2 diabetes and what we know about human beta cell proliferation. So this is a slide from Peter Butler's group at UCLA now 15 years ago. And it makes the point that even if you had type 1 diabetes, you can see my laser pointer okay, right? See that?
Yes.
Yes.
Even if you've had type 1 diabetes in this study 50 years, but as I'll tell you in a moment, 60, 70, 80 years, basically everybody still has some beta cells left. So this is an insulin positive cell out in the exocrine pancreas. Here's a few others. Here's a sick looking hyalinized islet, but it's got two notable things. One is there's no inflammation going on, and there are a bunch of beta cells in this sick looking islet. And then over here in a cross section of a pancreatic duct, there are insulin positive cells. Some people think those are the precursors for beta cells. So this is presumptive evidence for beta cell regeneration, even if you've had diabetes, type 1 diabetes for decades. More recently, this paper just came out last summer by George King at Joslin and Susan Bonner-Weir, who's a beta cell immunopathology person. And they make the point. Well, first of all, let me just tell you what Joslin medalists are. If you've had type 1 diabetes for more than 50 years at Joslin Clinic, they give you a medal. And they're proud of having started insulin therapy there, but they're also using this as a way to keep track of people who they've taken care of over the years. And so they also enroll them into life studies so that if somebody dies, they ask them to donate their pancreas. So in this paper that just came out recently, they did 68 postmortem exams on people with type 1 diabetes for more than 50 years, and some of them were 70, 80 years of type 1 diabetes. And basically the take-home message from this is 100% of people, even if they've had type 1 diabetes for many decades, still have some beta cells. Sometimes, as in this panel, they're scattered throughout the exocrine pancreas, as I showed you in a peroxidase stain a moment ago. Sometimes there are insulin positive cells in an islet. So red here is insulin. And then in some places, there are islets that are completely normal looking. And so just to remind you that the pancreas in type 1 diabetes looks like the skin in a person with vitiligo. It isn't totally homogeneous any more than vitiligo is totally pancutaneous. There are lobes and lobules in the pancreas of people with type 1 that are completely normal. No one understands that. And I'm not going to say more about it now, but I just want to leave you the point that 100% of people with type 1 diabetes, even if they've had it for 50, 60, 70 years, still have some beta cells. So if you, again, this is a Peter Butler study, if you measure beta cell mass in people with longstanding type 1 diabetes, in this study, it looks like it's something like maybe 5% of normal. There now have been lots and lots of these studies, and probably on average, people would say it's maybe 10% or so of normal. So beta cell tank is depleted, but it's not empty, and you can even say now it's never empty. And so this is obviously not enough to maintain glucose, not enough to give you enough insulin secretion to normalize glucose, but there's still something there. Are they functional? So this is a study from Richard Oram, who's at Oxford. And basically what they did is they used the newer generation, newer in 2014, which is to say the current generation of C-peptide assays. Prior to that time, the normal range went down to 200 picomolars or 0.2 nanomolar. So most people were negative. So in this study, they used the newer generation of C-peptide assays and gave people with type 1 diabetes for the durations shown at the bottom of the slide, gave them mixed meal tolerance tests and looked at their C-peptide response. And so the remarkable thing here is the majority of people with type 1 diabetes not only have islets, the majority of them, and even if they've had type 1 for many decades, still can mount something of a C-peptide response. So that suggests that these beta cells that you've been seeing in these pancreas sections are to some extent functional. So just to leave you with one point, on the left-hand side of the slide is type 1 diabetes, 2 million people-ish in the US. And the beta cell tank is depleted, but it's not empty. There are still beta cells. So similar studies have been done in type 2 diabetes. I don't have time to go through them in detail. I just want to leave you with a couple points. If you look at non-diabetic people who are lean, beta cell mass is in this range, call it 2% of pancreatic area. If you look at people with type 2 diabetes who are BMI, gender, age matched, their beta cell mass is depleted by about 40%. They have about 60% of normal. If you look at obese people with type 2 diabetes, TTDM is type 2 diabetes, beta cell mass is reduced compared to BMI, gender, age matched, non-diabetic obese people. And the beta cell tank, you can say, in type 2 diabetes and obese people is depleted to the extent of about maybe 60%. And these are metabolic syndrome, impaired fasting glucose people. Basically, we don't have time to go into why this is either. There's a component of beta cell death probably, but more likely it's genetic endowment. So if you're born and achieve this beta cell mass in childhood and you get 5 pounds overweight, you're likely to get type 2 diabetes. If you develop this beta cell mass through genetic endowment during childhood development, you can become very overweight and very insulin resistant and not get type 2 diabetes. So that's sort of where we are. The point of this slide is that everybody who has diabetes, whether it's type 1 or type 2, needs more beta cells. So what do we know about beta cell regeneration? So this is beta cell proliferation on the y-axis, and then age in human studies in embryologic life, neonatal life, childhood, adolescence, and adulthood. And so it turns out that human beta cells can replicate. They do replicate in the first year of life, and the proliferation rate is about 2% per day. That ends fairly quickly after childhood. And so by the time you're an adolescent, beta cell mass is reduced, proliferation is reduced dramatically. And by the time you're an adult, it's basically turned off or almost completely turned off. Some would argue there's still a little proliferation. So this is useful because it tells us a few things. If you wanted to make beta cells proliferate in a physiological way, you'd be shooting for about 2% per day. It also tells you that in the beta cells we get to study from NIH and private companies that sell human islets, we're sentenced to studying islets that don't replicate, won't replicate, and can't replicate. It also tells you that if you're going to do clinical trials with regenerative drugs, you're going to have to focus on this population. We're not going to be using experimental drugs in 1-year-olds or toddlers. So the good news is we know that they can proliferate, they did proliferate, and here's the physiological rate. Here's the challenge. We need to take these cells that can't, won't, never have proliferated and make them proliferate. So that's sort of what we set out to do. So that's background. So now let me just tell you about Harmine and drugs related to Harmine that we and others have called harmalogs. And again, this is review, but I just need to show you this to set up what's coming. So this is a study we published in 2015. The first author is Peng Wang, who I think all of you know is the brains and energy and driver of everything good that comes out of my lab. So Peng and I worked together in Pittsburgh. We had an idea of how we could develop a high throughput screen, robotic screen to discover drugs that would make beta cells, human beta cells regenerate. But we couldn't do it in Pittsburgh because we didn't have robots and chemical libraries. So part of the reason I came here was to do this. But we had made the cells that we wanted, that they needed to do this with in Pittsburgh and brought them here. And the bottom line is we discovered this drug called Harmine. It inhibits a kinase enzyme called DYRK1A, and it leads to human beta cell replication. So this is the first small molecule drug, or any kind of drug, that could make human beta cells regenerate. And again, without getting too technical, I'm just going to show you what we did to do this. So hang on with me for a couple minutes. So this is the regulatory region of the MYC gene. We all know that MYC is an oncogene when it's expressed at crazy high levels, but you may not know that MYC is actually required for every single cell in the body to replicate. So if you knock out the MYC gene in a mouse, they never get past the 2-cell stage. So if you want a cell to regenerate, it has to have MYC, and it doesn't matter what cell it is, it's every cell in the body. So every cell, every gene, so the gene is shown as a linear thing here. This line is the gene. And then this part here is the promoter. And so you can see that on the MYC promoter, there are sites where every mitogenic pathway you could think of impact. So here's STAT signaling, here's NF-kappa B, here's glucocorticoid receptor, this is cyclic AMP, et cetera, et cetera. This is Wnt signaling, et cetera. This is TGF-beta signaling, BMP signaling. So anything, our theory was, anything that could make that could activate MYC could make beta cells replicate. And so we could use this promoter as a way to detect anything that hit a cell and would activate proliferation. So think of this promoter as the on-off switch. We were just looking for anything that would tickle the promoter of the MYC gene in cells that we were playing with. Downstream of the promoter is the coding region. So this would be the first exon coding region of MYC. But we didn't want to turn on MYC. We wanted to turn on something that would give us a signal when we were driving MYC. So instead of using the MYC gene, we replaced that with a gene called the luciferase gene, which is the gene that fireflies use to give off their flash of light. So we had a promoter on a piece of DNA. We replaced it with a firefly luciferase. And then that enabled us to throw in any number of chemicals we wanted to at cells that had this. And if one of them would activate MYC, you'd see a flash of light. So basically, we made these cells, we fed them into the robots screened 100,000 compounds, and we got lots of hits. We actually got a bunch of hits that activated this. Then we had to sort through them and see which ones really could, which of those drugs could really make beta cells replicate. So we screened 100,000 compounds. This was in actually in Annenberg 18, actually. And at the end of that, we had one drug that worked, and that drug was Harmine, which is shown here. So Harmine is a small molecule. It's the same size as aspirin or testosterone or estrogen or whatever. And it fits into this little groove in this kinase called DYRK1A. So DYRK1A is a kinase. That means it takes a phosphate from ATP and sticks it on a protein. ATP normally occupies this groove or this pocket in DYRK1A. And so if Harmine is sitting in that pocket, ATP can't get in. And so through that mechanism, Harmine turns out to be a DYRK1A inhibitor, okay? So how do we know it makes beta cells proliferate? So if we take human islets and treat them with vehicle, there's no proliferation. This is BRDU labeling. But if we treat them with Harmine, we can see beta cells proliferating. We know they're beta cells because they're green and they're staining for insulin. And we know they're proliferating because they're taking up this thymidine analog called bromodeoxyuridine. And we can also see it other ways that I won't go into. And so it turns out that human beta cells can be induced to replicate with Harmine, and the proliferation rate is around 1.5%. Okay. So how does it work? And again, I apologize for biochemistry, but I just want to show you this because it sets up what comes later. So there are transcription factors called NFATs. They're normally sitting in the cytoplasm, unable to get into the nucleus, but with calcium entry into a beta cell or any other cell type, they're able to move into the nucleus and sit on promoters and activate genes that drive cell cycle progression. Okay, so if you take a beta cell, throw calcium at it, NFATs go into the nucleus, they activate these genes that cause proliferation. If that's all that happened, beta cells would be proliferating all the time. So the job of DYRK1A is to rephosphorylate these NFAT transcription factors and kick them out of the nucleus. So at a higher level, you can think of DYRK1A as the brakes on proliferation. DYRK1A is the brake on beta cell proliferation. So it makes sense, therefore, that if you inhibit DYRK1A with Harmine or other drugs like it, that takes the brakes off and you're allowed to get proliferation, okay? So it's complicated and not what you guys are used to thinking about, but I just want you to think of Harmine as taking the brakes off. It's taking your foot off the brake and allowing the car to roll or allowing cells to proliferate. So now what I've shown you is this is what we did when we were 1 year old. Our problem is we can't make beta cells replicate. Harmine actually does induce human beta cells to replicate and does it at a rate that's not far off from what we were doing when we were 1 year old. So when we reported this, we were anxious because lots of people had reported things that they claimed could make beta cells replicate, but other labs were unable to reproduce it. So after our stuff came out, several other labs have now repeated this. This is now, this is an old story at this point, but people in pharma and biotech at other universities like Stanford and Penn and Vanderbilt, et cetera, Joslin, Broad Institute have repeated this. So the point of this slide is the world now has a class of drugs that are able to make human beta cells regenerate. So we're in 2015 now. So that's good news, but is 1.5% a day enough to get you beta cell mass up to normal in type 1? And what about type 2? Well, in type 2, you're trying to get from here to here, and at 1% a day, it's going to take you a long time. Maybe over a few years, it could do it, but it's not going to be quick. In type 1, 1% a day or 2% a day, it's going to take a long time to get up here. So with that thought in mind and knowing that we can make beta cells replicate, we began to think of other things. We wanted to get higher rates of beta cell replication. And also, DYRK1A is ubiquitous. Every cell has DYRK1A. So we were concerned that if we just threw Harmine gave it to people or animals, that beta cells will proliferate, but other things might proliferate as well. So the next challenge is we're trying to come up with higher rates of beta cell replication and coming up with better beta cell specificity. So the next story is one that began with a grad student named Courtney Akefi, who stayed on as a postdoc. And so going back to this diagram, I told you that Harmine is the brakes on proliferation. So to use the automotive analogy, there are two ways to make the car go. One is to take the brakes off. And the other is to pour more gas in the carburetor or step on the accelerator. And so far, we've only talked about this part of this pathway. But what would happen if we threw a lot of calcium into beta cells or activated a lot of calcium entry in beta cells? Then maybe we'd have more calcium, more NFAT signaling, and no brakes, right? So could we rev up the engine and take off the brakes at the same time? And also, could we do it in a way that only happened in beta cells? So the model that I had was, let's do this and see if we can activate calcium inside beta cells. So here's what I showed you a moment ago. Now let's put it in a beta cell and think of things that could activate calcium. And so GLP-1 receptor agonists do that, sulfonylureas and meglitinides do that, some GABA drugs do it, glucose itself does it, and glucokinase activators, et cetera, and then other things that are less specific for beta cells. So Courtney's project was to basically take drugs in all of these classes and add them to Harmine and see if she could get more beta cell replication. And it turned out it worked. And the class of drugs for which it worked was GLP-1 receptor agonists. So this is back to your world. So these are drugs that you guys know about and use on a regular basis. They're all GLP-1 receptor agonists. And on the bottom half, DPP-4 inhibitors. They're all on the market. There are about 30 million people in the world taking these drugs every day. And it turns out that they act in part by increasing intracellular calcium, that they do other things as well. So we said, well, what Courtney discovered was that you could combine GLP-1 with Harmine and get more proliferation. So this is Courtney and again, Peng. And so this was published actually just this year, a few months ago. And Courtney's first experiment was, let me take human islets and I'm going to measure proliferation in beta cells. And so these are examples of Cas67 staining in beta cells. So this is what we call proliferation. And so let's throw either just vehicle, nothing, diluent at beta cells, or GLP-1, or Harmine, or the combination. And each one of these lines is a different set of islets from a different organ donor. And you can see that they don't replicate on their own. And if you give them vehicle, nothing happens. GLP-1 and all the other drugs in the GLP-1 receptor agonist family don't do anything to proliferation. Harmine did its usual in this group of patients, around 2% or 2.5% proliferation, similar to what we'd seen before. But if you take in these very same islets, Harmine and GLP-1 together, now suddenly you see much higher rates of replication. So you see replication rates that are up around, between, say, on averaging about 8% or 7% or something. Some of the organ donors, beta cells proliferated at much higher rates as well. So this is a rate of replication that actually clearly is an improvement over Harmine alone, and has therapeutic legs, I guess I'd say. So this is a dose response curve of GLP-1 in red bars looking at proliferation. Here we just use one dose of GLP-1. Now we're taking a big broad range of doses, and GLP-1 at any dose you choose doesn't drive proliferation. Harmine gives you, again, about in this study, maybe 2.5, 2.8%. And then these same doses of GLP-1 that didn't do anything on their own, when you add Harmine, suddenly you see this beautiful dose-related increase in beta cell proliferation, averaging in 6, 7, 8%. So then the next obvious question, if you're an MD, is like, well, nobody uses GLP-1 as a drug. What about the drugs that are on the market? So that's what this panel is showing. So this is exenatide, liraglutide, lixisenatide, semaglutide. None of them drive human beta cell replication. These numbers are the numbers of human islet preps they were tested in. Harmine did its usual 2.5%, which here is normalized to 1.0, one-fold. And you can see if you add Harmine to these same drugs in the same islets, under the same conditions and same labeling, et cetera, the proliferation rate is now twice as high, and it averages around 5 to 6%. So suddenly we have some beta cell specificity and much higher rates of replication. So to put this into context of what I've shown you before, here's what we did when we were 2. Here's what we're doing now. This is what Harmine did, and this is what Harmine plus GLP-1 receptor agonists do. So suddenly we're talking about therapeutically relevant rates of replication. So if you're proliferating at that rate and you have type 2 diabetes, it wouldn't take you long to get up here. Or if you start here, it wouldn't take you long to get here. And if you're starting at 5%, it would take some time, but not an unreasonable amount of time to get beta cell mass back up to normal. So suddenly we have a combination that gives you enough beta cell replication that in theory should be useful for everybody who has type 1 or type 2 diabetes. Okay, so that's nice that we can take human cadaver islets, play with them in petri dishes and microscopes, and see the beta cell proliferation markers go up. But do we really get increases in beta cell mass? And do we really get improved beta cell function? And can we actually see that we have more human beta cells? So I should tell you that intuitively, if not explicitly, right now we can't measure beta cell mass in people. You can at autopsy, but it's not a useful therapeutic exam. So you can, there's no way to measure beta cell mass in people. Similarly, there's no way to measure beta cell mass accurately in a living mouse. So what I'm going to tell you now is I'm about to show you is a paper that is online in a online journal called bioRxives, and we're finishing. This is the work of Sarah Stanley and Adolfo Garcia Ocaña in their labs. And so this is the name of the paper. This is the citation for it if anybody wants to read it. And I'm going to show you what it shows. And also I should acknowledge Carolina, who works with Adolfo, Alex Alverson, who works with Sarah, and Peng, of course, who does everything in my lab. So here's the idea. Let's take some human islets. We'll transplant them into skid mice that have no immune system, so they won't reject them. And we're going to put them in the kidney capsule. The kidney capsule turns out to be easy to exteriorize. So you create a little flank incision, pop out the kidney. You can take human islets. You can see them inside this catheter. pop a hole in the kidney capsule, squirt the islets in, withdraw the catheter, and the islets are now sitting in the kidney capsule. Then you can pop the kidney back in, staple the skin closed, and now this immunodeficient mice can live indefinitely and can be treated and can be treated with drugs. And so now we have in vivo human islets. Adolfo's been doing this for years, but to measure beta cell mass at the end of the experiment, you imagine, until recently, you'd have to just slice the whole kidney up and take histologic sections of thousands of microscope slides and try and guess what beta cell mass was. It's not time efficient. It's really not technically possible, and nobody does it. So prior to what I'm about to show you, there was no way to measure what happens to these islets over time. So then we developed a way to deliver drugs, implant them behind the shoulder blades of these mice, and then treat them for a month or for 3 months with these mini Alzette pumps. And so then at the end of the experiment, you can harvest the kidneys. You can basically fix all the tissues in the animal, harvest the kidneys, and the kidneys come out looking like this. Then they're opaque, so you can't really do any optical things with them. So you need to clarify them. And this is something that Sarah was doing routinely in brain that we didn't know how to do. And so she taught us and also Carolina in her lab how to clarify tissues. This meant that now you can see through them, you can do microscopy on them, and you can label them with antibodies, just as we do with conventional immunohistochemistry. And now there's a technique called light sheet microscopy that just takes serial planar sections through tissue, intact tissues, and then it creates images and basically volumetric videos, if you will, of in our case, beta cell mass, but most people are using this in brain to look at various brain nuclei. So here's an example of what Sarah and Adolfo did. I got to get out of this so I can use my pointer. Hang on a second. Where's that? Okay, so this is a mouse kidney, just like I showed you. It's been stained with antibodies for insulin and for actin. Actin just is made everywhere, so it's a way to show you the entire kidney. And so what you can see in 3D now is the entire kidney with the glomeruli in it, but you can also see the beta cells that were transplanted under the renal capsule. And this is a video. That means we're talking voxels. That means contained in the information in this video is actual beta cell mass. So this enables not only visualization of the islet, human islet graft, but quantification of the mass of beta cells. And so in the next slide, these are grafts from animals treated for either 3 months with just vehicle or 3 months with Harmine plus exenatide. So here's what a graft looks like in an animal that got 300 islets, 300 human islets. So you can see, 3 months later, the graft is still there. It's the same size as it was when it was put in. And then here's one that got Harmine and exenatide for 3 months. You don't have to know anything to see that beta cell mass is dramatically higher in the ones that got Harmine and exenatide. So you can quantify this, and without going into details, this is a summary of what Adolfo and Carolina and Sarah found. So this is the mass of beta cells that were transplanted, 300 islets, but in beta cell volume, this is the volume that was transplanted. If you do nothing, just treat them with vehicle. Let me get my laser pointer again. If you do nothing, a lot of the islets die in the first day after transplant. So at 1 month, if you harvest the kidneys, or 3 months, beta cell mass has actually declined. If you treat them with exenatide, as expected, because it doesn't drive proliferation, nothing much happens. If you treat animals with Harmine alone, beta cell mass goes down initially, presumably because they're ischemic and dying, but it begins to go up again. And at the end of 3 months, beta cell mass is higher with Harmine treatment than with vehicle or exenatide. But if you use Harmine and exenatide together, you see this dramatic increase in beta cell mass. This is a quantification of the video that I just showed you. So you can spin this different ways. If compared to here, from 0.2 millimeters cubed up to 7, that's about a 3.5-fold increase. Or if you compare them to the controls that were treated in other ways, it's about a 7-fold increase. And I don't have time to go into it, but part of this is proliferation. Part of it is making the beta cells function better, so they're more easily detected. They're making more insulin. There's a survival effect, and there may be other effects as well. But I just want to leave you with this part of the talk, saying that it's now possible to increase beta cell replication and beta cell mass in human islets in vivo in 3 months to rates that have clear clinical application. If you increase beta cell mass here 7-fold, it would be too much. We'd have to give lower numbers of drugs or shorter treatment to fix type 2 diabetes to get from here to here. For type 1, depending on where you think that the starting line is, let's call it 0.1, to get from 0.1 to 1.2, a 7-fold increase wouldn't be quite enough, but it'd be a step in the right direction. So either with more potent drugs or longer term therapy, you could get back to normal in type 1. In type 2, there's no autoimmunity. And so really all you have to do is fix this. In type 1, we still have to worry about type 1, about autoimmunity. We don't know what would happen in human autoimmunity. Would the human immune system go after the new beta cells? I'm guessing yes, but we don't actually know that. And so my thought is we'd have to use Harmine, exenatide, plus some sort of immunomodulatory therapy in type 1 for this to work. And we can talk about that in questions. So the next question is, what is Harmine anyhow, and could you actually give it to human beings? So Harmine is a small molecule that's derived from a plant in the Middle East that's called Syrian rue, or in Arabic it's called harmal. So the name Harmine comes from its having been extracted from harmal. In Brazil, there's a vine called Banisteriopsis, and in the Amazonian jungle and the Andes, people extract this vine, create an infusion called ayahuasca, and drink it. And both in the Middle East in historical times and in the Amazon in contemporary times, people drink this to get high. So this is our paper, Peng's paper on Harmine came out in Nature Medicine in the first week in March in 2015. Nobody called. About 3 weeks later, this came out in the New York Times. And basically, these are inmates from Brazil. They're the worst kinds of prisoners. They put them out in a jungle, a jungle prison that you wouldn't want to escape from because you'd get eaten by creatures around you. And if you give them a little ayahuasca, suddenly they turn into docile, happy, friendly, wonderful, nice people. So it makes the point. So of course, our phones started ringing off the hook from people with type 1 diabetes who wanted us to send them to Brazil to get this treatment to make their beta cells grow back. But anyhow, for me, it's just an illustration that Harmine may or may not have off-target effects that we do or do not want. So if you're interested in reading more about this, this is a nice story in the New Yorker a couple of years ago that describes what this ayahuasca trip is. The good news is it's a fantastic therapeutic, wonderful high. The bad news is if you overdo it, you get simultaneous enormous diarrhea and vomiting. So it needs to be dosed carefully, if you will. But really the scientific point is we don't know whether Harmine does this stuff or not. And so there is in the vegetable unit in Sao Paulo in the Center for Medical Studies, as well as people from the US and around the world, have done studies. They've gone to Brazil, they've had shamans make ayahuasca, they've taken it home and they've analyzed it. And I'm just going to show you the results of really one of maybe 2 studies on this. So we're talking journal. It's a journal you may or may not read on a regular basis, the Journal of Ethnopharmacology, 1999. And so they took 14 volunteers. They had a shaman make the infusion or the tea. The body weight of these people was 74 kilos plus or minus. And it turns out that they had shown, and others have shown, that ayahuasca contains other things. This is dimethyltryptamine, which is a potent and widely recognized psychoactive drug, neural stimulant, hallucinatory drug. This is tetrahydroharmine, which is a degradation product. This is Harmine, which is a slightly different version of Harmine, but it's an MAO inhibitor. And then this is Harmine. And so they could take this infusion, figure out how much of these things was in them. And so they realized that people who were getting this infusion were getting 250 milligrams of Harmine and 35 milligrams of dimethyltryptamine and some other stuff. So they did pharmacokinetics on these things. They measured Harmine. So here's Harmine going up in the first 15, 20 minutes, peaks at around just under 2 hours, and then it tails off. This is tetrahydroharmine, which is a metabolite. It goes up more slowly and lingers on. And then this is dimethyltryptamine. So dimethyltryptamine is the putative psychoactive agent here, and it's much more potent than Harmine in its psychoactive effects, if Harmine even has psychoactive effects. And so the psychoactivity is believed to be due to the dimethyltryptamine. But really, this is the world's literature on the use of Harmine in human beings, and it's sort of uninterpretable from a safety standpoint for Harmine in terms of what levels you can achieve and how safe it is. So with that in mind, we've actually started with planning and have submitted a grant to NIH on doing a phase 1 trial with Harmine. We've submitted an IND to the FDA. It's at NIH. Actually, it's being reviewed today and tomorrow. So next week I'll know. We've got a chemical synthesis group at the University of Minnesota who's making us FDA quality Harmine, packaging it in 100 milligram pills. And we're basically waiting to hear from NIH and the FDA as to whether, and the IRB, by the way, to see if they will allow us to do it. We here, I should say, is Dennis Charney and his, Dennis has done the ketamine story together with a guy named James Murrow and Manish Jha. And so there's a clinical research unit in psychiatry. We elected to use that because we thought that if there are side effects, they're likely to be psychiatric, psychological side effects. So we wanted people with that expertise. So that stay tuned. That's about to happen. We hope, depending on funding and FDA approval. So okay, we're not done yet. I showed you that Harmine plus GLP-1 gets you nice increases in beta cell mass, but we could probably make better, more potent DYRK1A inhibitors. So, we could do to exenatide what semaglutide did. We can make better versions, longer acting versions, et cetera. And then this issue of whether we need beta cell targeting or not. We don't really know if we have to target Harmine to beta cells or not. Maybe in therapeutic doses, such as we did in the mouse models with Adolfo and Sarah, we didn't see proliferation in any other tissue. We didn't see histologic abnormalities in any other tissue. We submitted them all to the veterinary pathology group. We haven't seen an off-target effect. So it's possible that we don't have to target Harmine to beta cells. Its effects on beta cells are somehow specific enough, and with GLP-1 together, that gives you some beta cell targeting. On the other hand, maybe we should be making targeted versions, beta cell targeted versions of Harmine. So we're fortunate at Mount Sinai to have a guy named Bob DeVita, who actually I'll show here. This is Bob. And so over the last few years, 5 years or so, 7 years, we've worked together on trying to make better versions of Harmine or other DYRK1A inhibitors that are more potent and more beta cell specific. And I want to acknowledge Kunal Kumar and Shalata Subaswang, who are MedChem people, research scientists who work with Bob over in the Icon building. So when I came, I said to Bob, it turns out his brother is a friend of mine from Pittsburgh. So he came in to say hello. I didn't really know him. He said, what do you do? So I told him the story. We found this molecule called Harmine. It's a drug that makes beta cells replicate. Turns out that Bob was the director of medicinal chemistry at Merck. And he looked at this and said, that's not a drug. That's a molecule, but we can make it into a drug. So Bob basically led the MedChem group at Merck for a few decades. And so partnering with them, we've now made literally hundreds of these molecules. So our lab meetings look like this. Here's the pocket that I showed you in Harmine. Here's this pocket in DYRK1A that Harmine fits into. Here's Harmine scaffolds, Harmine backbone, but maybe we should put some stuff over here. Maybe we should put some stuff over here. Maybe we should put this here. Maybe we should do both of those. And the concept is if you get a molecule that fits more tightly and more specifically into DYRK1A, it's going to be more potent and be a better DYRK1A inhibitor. So our lab meetings look like this now. Bob and Kunal and Shalata have synthesized hundreds of various Harmine analogs and now other scaffolds and other backbones that have nothing to do with Harmine, but that are potent DYRK1A inhibitors. And so one of the things we're trying to think about is if we've got this MedChem group and we have potent DYRK1A inhibitors, maybe we could hook them up to some molecule that would target them directly to beta cells and avoid every other tissue in the body. Clearly, we don't have that. If we had that, then we would be doing beta cell scans on people by taking such an antibody or drug and labeling it with indium or whatever, and be doing beta cell scans on people with diabetes or prediabetes. So we don't have that now. But GLP-1 is reasonably beta cell specific, and there are some monoclonal antibodies around now that may be beta cell specific, and we're playing with. So our thought was, let's see if we can take DYRK1A inhibitors that Bob has made, put linkers on them, and then be able to conjugate them or attach them to targeted delivery molecules. So conceptually, what we're saying is, you're a beta cell, and we know we have a special package for you. We're Amazon or UPS, and we want to deliver it to you, but we don't know your address. So we need a targeting mechanism. I put a missile on here. Bob is offended by that. He thinks it should be a UPS truck. But anyhow, so here's the idea, and here's one that Bob has actually made. So this is Harmine. This is the pocket. This is a nitrogen in Harmine that sticks out towards the air. And so one of the molecules they made, so the blue here is Harmine, the yellow is a Harmine analog that's got a linker on it that faces out into air, or actually into water, I guess you'd say. And it's got reactive ends on it that we can hook on to anything like GLP-1 or like a monoclonal antibody. And this particular molecule that we call T2C is published, actually. It's published in one of these papers a moment ago. I don't remember which one it is. It's this one. It turns out it's also been engineered so it can't get into the central nervous system. So if it has CNS effects, it actually shouldn't have them, and in rodents doesn't have any CNS effects. So where we are right now is we have molecules that we could attach to delivery molecules. So for example, we would take Harmine and attach it to exenatide or a monoclonal antibody. I haven't showed you, but we also have a TGF-beta story. So TGF-beta inhibitors also synergize with Harmine to give you dramatic increases in beta cell mass. And so Kunal has made a bunch of TGF-beta inhibitors with linkers on them that we can attach to things. So our overall strategy is let's try Harmine by itself. Maybe we don't need targeting at all. And so that's the point of the phase 1 trial. On the other hand, maybe we do need targeting. So our contingency is to make targeted versions of Harmine and TGF-beta inhibitors. So in wrapping up, or at least beginning to wrap up, because I still have some more to say, I've shown you the type 1 and type 2 result from inadequate numbers of insulin-producing beta cells. You knew that about type 1. You may or may not have known that about type 2. Most people with diabetes have residual beta cells. It could be stronger than that based on the Joslin study. Essentially, everybody with diabetes still has residual beta cells. None of the currently available diabetes drugs, and there, I don't know, you guys, again, would be fun to tally up the number, but the number I throw around is around 30 or 40 different diabetes drugs on the market now. None of them do anything for beta cell regeneration, beta cell proliferation, or beta cell expansion. So even though the fundamental cause of type 1 and type 2 is loss of beta cells, we don't have any, we have no drugs that mitigate that. And it turns out that human beta cells can be induced to regenerate, and it can be done using several different classes of small molecule drugs. So I've shown you the Harmine story. I've shown you the GLP-1 receptor agonist story. It probably applies to Harmine plus DPP-4 inhibitors. We haven't really tried that in animals. And it certainly applies to the Harmine plus TGF-beta inhibitor story. So we have now lots of ways to achieve expansion of human beta cell mass. So the next challenges are identifying a perfect beta cell specific targeting molecule. The world doesn't have that now. We have ideas about how that could be done. We also, acknowledge, and the field, I think, acknowledges that maybe we don't really have to do targeting. The data that Adolfo has generated on off-target effects in other organs says there aren't any off-target effects. You just get beta cell expansion. So we need to do long-term preclinical safety studies. So that means we need to do 6-month studies in rats, 6-month studies in monkeys or dogs before anybody's going to allow us to use this over the long term in human beings. Our strategy will be something like this. With the phase 1 trial, you don't need a lot of preclinical data. We have enough preclinical data for that. We hope. We'll see what the FDA and IRB say, but we think we're okay there. But that would just be a 1-day study to figure out what dose of Harmine we could give safely to healthy humans. The next step would be to use it, in my view, is to move to people with type 2 diabetes who are taking GLP-1 receptor agonists, and then add Harmine to that and see if we can get a bump in C-peptide secretion from adding Harmine to a GLP-1 inhibitor, a GLP-1 agonist. But to do that, those kinds of studies, we're going to need long-term animal studies. They're expensive. If you want to do an FDA acceptable 6-month study or 3-month study in primates, we're talking millions of dollars. That's bigger than grants. So we're hoping that somebody in pharma or biotech would be interested in funding that kind of study. And then, as I mentioned, this should work nicely for type 2 diabetes. My own bias would be we're not going to do anything good or anything in particular for autoimmunity and type 1 diabetes. So that's going to have to be managed independently. So the future there for me looks like something like anti-CD3 antibodies, teplizumab plus Harmine GLP-1, but we're still a ways off for that. From a type 2 point of view, it's not relevant. You just need to make the beta cells retain differentiation and expand. That's good news for type 1 because we have artificial pancreas thanks to Carol and David and their teams. And now we're about to have beta cell regeneration. So that means the last chair that you have to kick out of the stool for type 1 would be managing autoimmunity. So for me, that's pretty amazing progress. And then I've told you the human beta cell in adults proliferate slowly, but I haven't told you why. And the short answer is no one really knows in great detail. But that's an area that we're working on, basic science-wise, and we actually know a lot about that. And it's pretty fun. It's pretty interesting, but it's still a black box. So is this even reasonable? I mean, what about the other things? Look, we already have pancreas transplant. We already have islet transplant. So those are both very efficacious. They work. The long-term, particularly with whole pancreas transplant, the long-term graft viability and diabetes reversal is terrific. The problem is it's not scalable because as you can imagine, there aren't enough organ donors to come up with enough pancreas to generate enough islets. The cost per patient would be crazy high. And immunosuppression has its own oncologic risks. So in organ transplant studies, people routinely get lymphoproliferative disorders and other solid tumors like squamous carcinomas. It's FDA approved, it's in use and reimbursable. What about stem cells? So we're about 20 years into stem cells. They've yet to make it into human trials that have shown efficacy. It's certainly not scalable. If we're talking, in the US, somewhere around 2 million people with type 1, it's not scalable to 2 million people. And it's certainly not scalable to 400 million people in the world with type 1 or type 2. The cost per patient for both of these will be exorbitant. And stem cells have their own risks of teratomas. And it's not in use because it's not approved by the FDA because they haven't passed the trials that they need to. In fact, the trials are sort of going back to zero and being modified, the actual encapsulation devices. In theory, you might be able to convert alpha cells to beta cells, and I won't say much about this. It's complicated. The bottom line is it's an idea, but whether it's doable or achievable isn't clear. So if you look at the stuff we're doing from that point of view, is this efficacious? Well, in human islets and mice, it's highly efficacious. We have no human data though. Is it scalable? Absolutely. I mean, hundreds of millions of people are already taking GLP-1 receptor agonists. No reason they couldn't add Harmine onto that. Cost per patient would be the lowest on the chart here. We don't know if it would be oncogenic. We get that asked all the time, and I'll say something about it in a moment. And are the GLP-1 drugs on the market? Of course they are. The DPP-4 inhibitors are as well. Harmine certainly is not. So are there examples of drugs that we use that are not particularly well targeted, that are mitogenic? Well, yeah, I mean, you guys prescribe them every day. And so the trick is figuring out the dose you want to use. I mean, you have to be lucky. It has to not have adverse effects in therapeutic doses. But if that's true, then what you need to know is how long can I give it safely, and what are the doses that I can give it in safely. So just to end, that's where we are with this. We're I would say pretty excited about this stuff. We filed a bunch of patents through Sinai on this. And we're little by little moving it forward. So let me end by just saying, first of all, acknowledging Pung, Adolfo, Courtney, Ezra Caricos, Don Scott, Dirk Homan, who you guys know, a bunch of other people. Let me just make sure. Here's Bob DeVita and people in his group, Roberto Sanchez, et cetera, and Shalata. And then we get islets from lots of places, including the NIH's IIDP, and the work that I've shown you is mostly funded by NIDDK, with a little bit from JDRF, JDRF is sadly suffering now because of COVID and financial crises. So let me stop there. I'd be happy to answer questions. Let's see.
So it looks like we have a comment and a couple questions in the chat. First from Dr. Levy, very exciting about the phase 1 trial. And she says she'll have plenty of type 1 volunteers who'll be interested.
Sorry, right, who said that? Carol?
Carol Levy.
Oh, good, Carol. Well, I mean, I was thinking of you as I made these slides. I'm still trying to find where are you guys? Okay, now I can see. So yeah, yeah, and also, I mean, to the extent that I mean, Carol, if this takes off, you're gonna be the first person I call for clinical trials. We need the psychiatrist to do the safety stuff, but I mean, my thought after that is let's move it into the endocrine division. I've talked to Carol. So NIH has this thing called TrialNet, which runs all their type 1 diabetes clinical trials. The woman who runs that is named Carla Greenbaum. Carla is a friend, and I've run this by her, and she said, look, I mean, if you can get safety data and dosing data, well, their mandate is they can only treat diabetes. They can't do phase 1 trials in healthy people. That's not what TrialNet's about. But if you can show safety data and dosing data, we would jump on this and use it in type 2 diabetes for TrialNet and possibly in combination with anti-CD3 antibodies, Metabolism. So Carol, you're going to be the first person I call. David, and if she doesn't answer, David and Emily, I'm calling you.
All right, so the next question is from, I think it's Louis, the violinist. Which immunomodulator would you use in combination with Harmine for islet expansion? Perhaps you tried NCI3, the NFAT-CAM antagonist drug used in autoimmune diseases and to prevent transplant rejections. Also, would anti-CD3 work in beta cells?
Yeah, so it's a good question. So NFAT stands for nuclear factors activated in T cells, NFAT. So what Harmine is acting on at the end of the day is NFATs. And their site of discovery was in T cells. There are papers in the literature saying that drugs like Harmine actually are immunosuppressive and favor T regulatory cells, which would be a good thing in type 1. On the other hand, I mean, that's a nice idea, but whether that actually translates to humans, we don't know. So a couple of things. I mean, I guess a fair summary of the immunology of type 1 diabetes is no one really knows exactly what's going on. It's still pretty primitive. The more you dig into it, the more complicated it gets. Having said that, there are now, I think, 5 different clinical trials, most of them involving TrialNet and Kevin Harold, that have shown that anti-CD3 therapy is effectively immunomodulatory in people with early type 1 and people, first-degree relatives at risk of getting type 1. People would argue over whether that's a long-term safety reasonable thing or not, but as the trials get more and more done, it looks like it probably could be. On the other hand, I think it's also reignited the hope that other classes of drugs with other targets in the immune system besides T cells might also be effective. So for me, the way to clinic for this is type 2 diabetes where autoimmunity isn't an issue. And the type 1 diabetes immunotherapy, immunomodulatory therapy world is going to continue to mature over the next few years. And by the time, hopefully, we can show that it works in type 2, then they'll be willing to, and I think excited to partner in type 1 as well.
So we're just at about 9:30, but it looks like there's a couple more questions. The first one is from Alice Levine. First of all, thank you for a great talk. And also, it looks like type 1 diabetics clearly have some low residual beta cell mass remaining, but have 0 insulin levels. Is beta cell regeneration equal to restoring normal insulin production, processing, and secretion? And then a follow-up, also with most endocrine hormones and enzymes, you only need a tiny amount to get full function. Why do you need so many beta cells?
So the first one, Alice, it's a perfect question. And the answer is, the only way I can answer that, well, I mean, Adolfo has done studies, and they were in that paper of Courtney's in Science Translational Medicine, where we gave streptozotocin to kill beta cells in the mice, transplanted them with human islets, and then treated them with vehicle or with a Harmine exenatide combination. And basically, those human islets, when treated with Harmine and exenatide, reversed diabetes in these type 1 models. So the answer is, I mean, what I'm excited about is there's no limit on the increase in beta cell mass we can get, and they function normally. I haven't shown you that, but we have tons of data showing that not only are there more beta cells, they're actually better differentiated than normal beta cells. And the second part of the question, can you just read that again, Emily?
With most endocrine hormones and enzymes, you only need a tiny amount to get full function. Why do you need so many? Why do you think you need so many beta cells?
So that's sort of a teleology, evolutionary. Well, here's what I think about this. I mean, actually, if you think about beta cell mass in us, it's pretty tiny. Total beta cell mass in a human being is 1 cc. That's how many beta cells we have. You take 1 million islets in the pancreas, and they're 40% of the beta cells. That's how many beta cells we have. So it's way smaller than thyroids or adrenals. And so my view is that there's immense evolutionary pressure. You got to have some insulin, but there's immense evolutionary pressure not to have too much insulin. You don't want to be hypoglycemic while you're running away from a saber-toothed tiger. And you don't want to get hypoglycemic and pass out while you're chasing a water buffalo for dinner. So in the world of lizards and proto-humans, too much beta cell mass and too much insulin is lethal. Whereas in a world of starvation, you just need a little insulin. So I think we have, evolutionarily speaking, exactly the right number of beta cells, although you can see that different people have different numbers of beta cells, right? I was showing that in that type 2 sort of summary slide. And that's almost certainly genetically endowed. So, I think we actually have a small number of beta cells in all of us. That's enough to protect us from getting hypoglycemic. And if we ate caveman diets, would be more than ample to make sure none of us got diabetic.
Thank you. Beautiful talk, Andy.
Thank you, Alice. Happened right here in Mount Sinai.
And then the final question from Dr. Genève.
Great talk, Andy, and great example of translational science. So wonderful. Many years ago, when thiazolidinediones didn't have any bad reputation, there were data that they also increased beta cell proliferation. And in humans, the only treatment for diabetes prevention with a legacy effect, that is, you keep seeing in normal glucose tolerance off the drug, it was the tripod study with trobutazone and former GBM. So have you thought of looking at TZDs and adding that into your cocktail?
Yes, hang on. So let me just show you a slide here. If I can find one more slide quickly. So, I mean, you've seen this slide before, some of you, but I mean, how do I make full screen? Can you still see my screen?
Yep.
So, human islets and rodent islets not only are different in many physiologic ways, they also architecturally are different. They're different in many ways. So I've shown this slide before, but oops, come on. this is a list of drugs that work in driving rodent beta cell replication. And I remember for sure, but I think TZDs are on here. Actually, I'm not seeing them, but you're right. They have been shown to drive rodent beta cell replication. To my knowledge, and I think we have tried them, but I'd have to go back and dig around. Maybe that's a good kick that I need to get us to go back and do that. But basically, these are all the ones that work in rodent islets. This is the list of ones that work in human islets that have been tested. So the short answer is I can't remember if we've done that, but it's a good idea. We should go back and do it. But again, so we're sort of, now we want selectivity. So I guess that would be liver targeted as well, right? So we'd like something that only talks to beta cells.
Well, I mean, in the tripod study, there was no hepatotoxicity, and the hepatotoxicity was probably specific to triglyceride, and there are, plenty of other people.
Yeah, but I mean the Harmine effects. So we'd be giving Harmine plus that, and they clearly have synergistic effects in beta cells. But yeah, and until we try, we don't know. So that's a good suggestion. We should do that. I hope Peng is listening. I doubt it. He's probably working in the lab, but that's a great idea.
Okay. Well, thank you very much. We need to get your annual update because you're making so much progress. Scheduled when we have the fellowship interviewees. It's really sad that they missed this.
All right, next year, hopefully we'll have more next year, Alice. Let's coordinate. But yeah, and I guess I just want to say that, you guys think of me as probably a lab geek who was born with a pipette. The truth is I was a GP. And so if you find this stuff complicated, I'm really speaking to the fellows and residents who might be there. This is all understandable. I didn't understand any of this stuff, 40 years ago, but after a couple of years of fellowship and playing around the lab, it's absolutely understandable. It's just another language. So don't think of this as intimidating. It's actually completely understandable, and serious scientists probably think I'm fairly unsophisticated. So it is what it is. All right, everybody. Thank you very much.
Thanks.
Thank you.
Okay.